Okay, my outstanding friends, this is going to be exciting for me at least. I have been studying diseases, viruses, infections, enzymes, catalysts, bacteria, proteins, all of these different things that are involved in the chemistry of the body. Because right now you are literally going to die in 5 minutes if you don't have chemistry going on in your body. And every single molecule in your body right now is dying. If it does not get invaded, literally invaded, within the next 5 minutes with oxygen and all of that stuff, and then dump all its bad stuff out, it will literally die. That's why you have to breathe. You don't have oxygen for 5 minutes, it's all over. So what is going on in these cells, and what protects them from invasion by viruses? And what is a virus? Where's the origins of virus? What is the basics of viruses? And I'm going to tell you right now, they know very little about viruses. This is, where did viruses come from? There's much debate among virologists about this. There's three main hypotheses, and those are only guesses. Good morning, my outstanding friends. This is Roger once again, and today we're going to go into research. And I am going to do this research with you because I am going into trying to understand what viruses are, where they come from, how they work, what do they do within your body, and is there anything we can do about them? I do research. I'm not telling you to take this or don't take that or anything like that whatsoever, so don't even think about that. What I am going to discuss and hopefully get some feedback on is virus. First of all, infection. How do they get into you? What are they made of? Are they alive? They don't know any of this stuff right now, and they still don't know it. I'm not kidding you. Are viruses alive? Well, maybe yes, maybe no is what they say. I'm serious. There's very little understanding of the difference between a virus and a bacteria and an enzyme catalyst, you know, the different types of genetic construction, basically, because this is just a genetic code. That's all it is a code. It says, do this job. And what is the job? Kill the guy. That's what the virus does. Well, how does it do that? And what is it attacking? Why are we having all this influenza and lung disease? And the rubbery parts of your body are going bad from viruses. Why? Why is that happening? Why does your heart go bad sometimes from viruses, viral infections? Your blood vessels crack and break. Your lungs are just collapse. Why? What is going on? This is what we're going to learn, or we'll try to learn. We're going to see if we can think of anything about it. Because right now, I'm going to tell you right now, everybody relies on the old science that they told them to say, oh, virus isn't alive. Well, no, there's all kinds of theories now, because people are starting to say, you know, let's stop and think a little bit. This is the problem. It's read page 25 through 35, and then you're going to know everything there is to know about viruses. No, that's just not the story. Nobody knew, and nobody knows now, because it's forbidden to really discuss this in great detail. And again, I am a researcher. I'm not telling you to do anything, but I am trying to learn about viruses, and you should too. Or if you're a doctor, you should pay attention to this. I'm telling you right now, if you're a doctor or a researcher, you will learn something by watching this. All right, this is what happens with influenza and these viruses. Now, what do? They damage the alveolar barrier by breaking through, basically, and disrupting the epithelial cell tight junctions. What is a tight junction? First of all, we're going to have to look into that. And what is this epithelial cells? All of that stuff. We're going to have to define what we're talking about. You can't just breathe through this. Now, they're talking about the abstract of this whole thing. This is a major cause of respiratory failure, of basically dying, a virus infection is damage to the epithelial barrier of the pulmonary alveolus, which is the barrier between the air and your body. All right, there's a barrier in there that says, let the air come through, don't let any other garbage come through. Well, that's supposed to be a tight junction. Well, these little tiny viruses are so tiny, they can squiggle through these tight junctions if they're not in perfect condition. That's what's called a pre-existing condition. You're ready to be invaded. Why? Why aren't your tight junctions tight? There's got to be a reason for it. One guy goes in there, he gets sick, and a couple minutes later he's fine. And another guy gets sick and he dies. Another guy goes in, he doesn't get sick at all. Why? Why did they get invaded? Well, we're going to go by. We're going to try to understand how the viruses enter the body first of all, and then what do they do once they get in the body, and are they alive? And I will tell you right now, they say it depends on how you define living. There's these particular viruses don't sort of exist on their own and have a life on their own. They need to be living within our cellular network. And they use our plumbing and all the stuff that we have. Like they move into your house. A cell is like a house. They move in, they take over. Now, they had to get there from somewhere. But once they get in, they claim, and this again, these are all claims to me at this point. I haven't done the research that they've done, so I can't dispute what they're saying. But I know what I can see and what I can determine and what the questions I want to ask. Let's go with that. These are the questions that I have. So that's what we're going to go through is the questions. And I'll be able to answer some of them myself, but some of them I'm going to need help with. That's what I want. I want some interaction. All right, so we're going to be talking about viruses. And almost everybody can, I got a virus, I got the flu, I did, influenza. It's associated to tissues that are in your lungs quite often. Now, let's just take a little biology lesson, an anatomy lesson of what the lungs in that network looks like. All right, here's the whole shooting match. When you breathe in, the air comes down your throat and branches off into these little branches. Now, those branches are inside the lungs, all right? And they feed all these little air tubes. Now, so what do you have coming down here? Air. And of course, all the other things that you're breathing, and smoke, and all kinds of things come in here. And then you cough them out with the phlegm. You make like phlegm, and it's like a gooey substance that you cough up every now and then to get rid of stuff. Now, this is what's inside the lung cavity. Is these little? Let's focus in a little closer on that one. All right, well, what are we looking at here? Every lung has a very, very thick coating on it. It's called pleura, and it's around here on the outside, very thick. And that coats this lung. Inside the lung is these lobes. Now, you shouldn't have any invasion through this barrier because it's a membrane, so that protects you, but that's inside your body, so normally that's not going to be an issue. But you're breathing in all kinds of things, pollutants and all kinds of stuff. So the tight junctions that are in these alveoli areas where the air that you're breathing in bumps up against the tissues that have to allow oxygen in, it has to. So you have to be able to permeate that barrier, but that barrier has to be tight enough to keep out these tiny, tiny viruses. The viruses are absolutely like a nanometer size, which is like atomic. All right, so you should have an idea of how it is. There's the air coming down. All the air floats through here. The blood circulates very quickly through your lungs and picks up the oxygen, and it spits out the carbon dioxide. And all it is there's two different levels of oxygenation. CO2 is carbon dioxide. All right, and that's got two oxygens. What you're going to be running through your body is iron oxides, which is FeO3. Well, FeO2 is the stuff you blow out through carbon dioxide. FeO3 is the stuff that absorbs in your body. It sucks up one of the oxygens, and that's how you breathe. You need oxygen. Everybody knows that. All right, so we know we need oxygen, and that's how that moves, and we know we need everything else in now, and it moves through the blood. But the blood at this point is only supposed to be able to take in oxygen, basically, that kind of a size of a molecule. And so that's my point. That's my figuring right now. I figure these tight junctions are the issue. Why would they be the issue? Okay, so here we are at the European Respiratory Journal again, and this can get pretty complicated, but I like to go for the easy route. I am a simple-minded person. What I am going to discuss is the damage, barrier damage, was associated with disruption of the tight junctions amongst epithelial cells, and specifically with loss of tight junction protein, caudin 4. Well, what is a protein, and why is it specifically related to this particular protein and tight junctions? I could tell you what proteins do, and we'll look into this particular protein. But proteins are chemistry sets. Let me show you. They are instructional chemistry sets. All right, like I said, I like to try to make things simple. Bacteria create strings of beads, literally magnetic beads, just like these. They're magnetic beads. And once they come out, they start to fold and twist and bend and do all these things, and they end up in a ball, like that. Now, what will happen is when they come out, they will start to twist. But watch what happens when you go near, you see? It doesn't want to be next to it. It will not. You can't get these two together. So what happens is when it comes out, it goes, and all these different things happen, and boop, all of a sudden it locks in, or boop, all of a sudden it locks in. But it has to be exactly, I'm talking about exactly that particular looking thing, which is basically what we're looking at here. And guess what that is? That is identical to what you're seeing here. And what is this? This is nothing more than a bunch of magnets. And what do magnets have? They have positive and negative attachment places. And what is that, Roger? Well, that just means that some of them will stick together, some of them will push away. If you have an exact copy of this, and I mean exact, with these little balls hanging off the sides and these little tips here and there, it'll come up and it'll find other chemistry that it knows it wants to attach to, and bam, it's called click chemistry. It will hit that other chemistry, boop, and instantaneously millions of the product that it just hit against, they'll all change instantaneously at the speed of light, because that's how fast magnetism works. That's what a catalyst does. Now don't get too worried about all these little terms and tricky things. It'll become very simple. A catalyst is an enzyme, and that's what an enzyme is. An enzyme speeds up a product's everything. But you can't get one of these without starting without one of these. Oops. Can't see any of this, can you? All right, you can't get this fancy stuff over here without starting about with a string of beads. And you can't get the string of beads without bacteria. Now, virus, they're saying, is not really a living creature that is like a bacteria that swims around and it's having fun on its own life. Who knows what it does on its own time. But when it's spitting out enzymes, it's doing this. But it's other than that, it's got a life of its own. Well, they say the virus is not that way. The virus has to be inside your body to be doing work. It's got to come from somewhere, though. This is where, and nobody knows where viruses come from. All this stuff is unknown, literally unknown at this moment. And they have no idea, they're not even doing any studies about how they affect your lung tissue. I'm serious. I just read that. It blew me away. Okay, this is that Claudin 4 that they're talking about, which is the protein we're going to look at. I'm a visual guy. I like to see things and try to think in my mind, how does this work? How did that thing invade this? And why did that thing turn to this different texture and so forth? But here, listen to this. This is about this Claudin 4 and epithelial cells and all that. And this is the big article they did from European Respiratory Journal. But it's from 2016. Now, I don't think they've done anything different since then. But it shows, here, look. However, to the best of our knowledge, no study to date has investigated whether the IAV can damage the AJC of alveolar epithelial cells. AJC is the apical junction complex, and it's where everything ties together. The tight junctions. There's no studies on it. Okay, are viruses alive? They don't really even know if they're alive or not. It says that's a good question. What is the understanding of what we mean by life? They are clearly capable of carrying out commands once they get in your body. How do they get in your body? How do they get into the cells? Well, they call it capsid encoding organisms. A capsid is like a little capsule. It's almost like a seed. And it appears that they're almost like seeds. Now, where they come from originally has to be some kind of seed-bearing organism. But once it gets into your body and takes over your cellular mechanisms, then it seems to be able to really take over. However, you still are going to be able to defeat it. And a lot of people, they get fine very quickly. So it's not something that, it takes over and it takes over your entire body. Some people it does. But they have no way to fight back. And how could they fight back? The only way they can fight back is with chemistry. Everything is done in your body. 100% of everything. 100% is chemistry. You breathe, chemistry. You urinate, chemistry. You eat, chemistry. Every single thing is chemistry. Not a single thing is not chemistry. So, what affects chemistry is enzymes. All right, just remember this. It says the prime characteristic of a virus is they absolutely have to be inside of a host to live. They cannot live on their own. Now, viruses need to carry enzymes with them. All right, they have to have some enzymes. Enzymes do all the chemistry. Without them, you get no chemistry at all. None, zero. All right, just to make this very simple. This is an enzyme. It starts out being a protein, the primary structure, which is squirted out from a bacteria. So they squirt out this string of beads. They come out like this. And then they turn into a secondary structure, which is these different types of molecular forms. And then they start to tangle up, and then they end up being the end product, which is an absolutely elegant molecule. And if you look at this one here, this is probably the best example. Right there. You have all kinds of pluses and minuses and all kinds of centers of negativity. They call them molecular moments. All kinds of polarities going on here in an extremely elegant pattern, and it's got to be exactly that pattern. If it's not that pattern exactly, if this one here was missing, it wouldn't work anymore. It wouldn't work anymore. It would be called denatured. Its nature would be destroyed. Now, what happens is these things here, they squirt out like this, and then they retangle. Now, you see this here? Whoops, you see it attached there? Now, if it came out in the other orientation, it wouldn't attach there. It would say, no, I'm not going there. I'm not going to get there. All right? Unless it hit exactly where it's supposed to. And that's what makes click chemistry work. These particular types of enzymes, when they touch the molecules that they are supposed to interact with, millions can be changed just like that. Millions in less than a second. by having the exact chemistry of molecules and polarities come up and go bang. Every other molecule in there changes instantly because it's traveling at the speed of light. So every other molecule that can attach to that particular magnetic signature changes. Done. That's what enzymes are all about. And we don't get these without bacteria. So if the virus has to carry an enzyme with it, somewhere there was a bacteria. Now, did the bacteria create an enzymatic little package that had some, because enzymes, they break through food too. They break through cell walls and break food open so you can get into the food. That's what enzymes in your intestines do to absorb food. So this is a tangled web. We have to start to understand, how do these get produced, which is from bacteria, and then what do they do, and how do they affect these tight junctions? And the reason they go through the tight junctions is because these viruses are so tiny. They're almost the same size as oxygen. They're so tiny. Okay, these are these very tight junction membranes. And this is about cancer and how you get invaded. And this shows the different tissues and how they, it's collagens that are in these tissues that are supposed to be in these membranes to protect you. Now listen, here's what it says. Claudins are a family of transmembrane proteins, which I showed you how the proteins are these big squiggly things, all really fancy, and they're magnetic, that are structural and functional components of tight junctions, predominantly in epithelial cells. Just precisely what we've been talking about. And if they're not working good, if they're loose, you're got a pre-existing condition. You're open for invasion. Okay, at the present, they don't even know how many. What they're trying to do is figure out how many human cells there are versus bacterial cells in the body. The bacterial cells make all the enzymes. This is what I would like to see happen. This is what I'm hoping to see. A viral study database. And it's just a database. No intervention at all other than collecting the bacteria samples from people. Fecal samples, spit, lymph nodes, we might be able to get them from autopsies, who knows. But 100% of the body chemistry, under your armpits, swabs here and there, and then just package it off and send it to a lab, which would run it through a process, which would be established. This would go here, this would go here, and out it comes, and then John Doe, such and such an address, and all these different things, your age, your sex, your race, your lifestyle, your foods, your region you live in, because you're eating and drinking other different stuff than other people, your basic hygiene, your social status, all these things can be taken into account in that database. And then what is the bacteria that lives in you? Fecal bacteria is predominantly what creates most of the bacteria in your body. But you're going to have stuff in your lungs, your lymph nodes, and all that stuff. As it evolves, it would be better. Now, what we want to do is get a database going. Nobody's getting anything whatsoever of medical advice or anything at this point. We get a database going of how many species of bacteria live in, say, me, and what their relative quantities are. And we compare it to the norm of the database of a huge ball of data. And eventually, you should see some patterns if this is true. If what I'm saying is true, you'll see a pattern. Everybody that has a certain type of disease, most of them are going to be missing a certain type of enzyme, which is a certain type of bacteria. Now, how can we fix that? There's several different ways to address it, but first of all, you have to figure out, what are you missing? You can't just shotgun approach this. So this is the only way we can figure it out, is to get a database of what is normal. Now, they did this, but all they did it for was how many is a normal number of cells in a human body. Okay, what they're trying to do is look into the microbiome, which is all of these bacteria that I'm talking about. But they're doing it sort of in the, I don't think the correct way, in my way of thinking. They say a first step towards understanding the bacterial communities that form the human microbiome and how they might influence health and disease. Take a census. Yes, exactly. What it lives where? Of where these communities are in your body and how much of each one there is. One way to accomplish this is to sequence these genes. No, that's not the way. Here's the way. Is to take samples. Collect bacterial samples. Fecal samples, lung, spit, lymph, 100% of the body. Where do these things live? Under your armpits, swabs, and so forth. We'll show chemistry. That chemistry will indicate what is in your body and what bacteria live there. Once you find out you're missing certain bacteria, you may find out that everybody that has a certain disease missing that bacteria. And if they're missing that bacteria, they're missing that enzyme. If they're missing that enzyme, the job that enzyme did never happens. And enzymes are the only thing at all that does any chemistry in your body. They are the facilitators of every single chemical reaction in your body. They're called enzymes and catalysts and so forth. Now, they protect you, they break down food, they create products, they do all the things that are required. They take out the trash, they move things around. So if you don't have the bacteria to protect you and to feed you, you are not going to do well. We need a database of all this, and then we can compare everybody's. You just do a swab of this, a swab of that, some feces of this, a spit in a cup, take a little urine, whatever you got, and put it in a bag, off it goes. The lab tests it, they send back, here's your results based to the whole world. And you need to have a little, you got to do a little work. And as you put your age in, your sex, your race, your lifestyle, your foods you eat, the region you live in, hygiene, type of hygiene you could practice, social status, everything. All of that stuff plays a part into how you absorb the elements that are around you. And sometimes the region of the country you live in is a real problem. Or it could be real beneficial. It depends. Everything's chemistry, 100% chemistry. If you're on this type of chemistry eating from there and this chemistry eating from here, two different results. I say we need a viral study database. Just wide open. And all you got to do is send in your stuff to it. Maybe you have to pay whatever it is, some kind of money, and then you send in your stuff, and then you get your result back. Here's how your bacteria fits to the rest of the world. You may just have to take some probiotics or something to make yourself fall in line with the norm that aren't sick. All right, because people that are going to be ill, I got a feeling you're going to find a lot of them are going to have the same bacterial analysis, because the bacteria creates the enzymes create the chemistry decides whether you're sick or healthy. Okay, there's something about the invasion that some people don't understand. It's the size of the particle depends on how easy it is to invade. There's something called tight junctions. I probably already mentioned them, but they are places where you should only be able to allow gases through and some very, very fine molecules. Because look at the virus, the size of the virus compared to blood cells. But you have to take in and out of things out of your tissues for your body to exist. You got to dump the trash and you got to put new stuff in. So it's the membrane that is the key. And the membrane is a product of collagen. Collagens create tight junctions or they can get loose. And when they get loose, that's when you get viruses and your lungs get all floppy and you can't even inflate your lungs anymore. You have to be intubated. So it's an attack on the collagen, I believe. All right, this is what I talk about collagen. It accounts for 30% of your body's proteins. the strength, muscle, skin, bones, connective tissues. It does virtually everything in your body. And they say they're lacking most collagen supplements. They don't understand it. The research they don't understand. But a well-balanced diet gives your body the raw ingredients it needs to help it make collagen naturally. Yes. However, those raw ingredients have to be worked on by some other bacteria that's in your body, enzymes. There's no other way around it. They don't just come in there and then turn into something. And the bacteria are there to break them down to make them digestible. It's the most abundant protein in your body. They're made from amino acids. And it's all your connective tissues, and these tight junctions are based around collagen. All right, this is it right here. Here's what collagens do. They help grow fibroblasts to form your dermis, which is your skin. And the middle layers of your skin is specifically what they're talking about, which helps new cells grow. Plus, it is where your membranes exist. It plays a role in replacing dead skin cells, collagens do, providing a protective covering for organs. Every organ is coated with collagens to separate it from their membranes, giving structure, strength, elasticity to your skin. It's the rubbery stuff that pulls, and it's also the rubbery stuff in your lungs that does that. And it helps your blood clot too. Now, there's 28 different types of collagens they're talking about here. They differ in how the molecules are assembled. Some of them are real stretchy, some of them are really hard and they don't move easily, some of them are really like springy, but that's the key to them. They're all different types. The cell components that are added when collagen is used in your body. They're assembled in your body. The collagen fibrils have at least one triple helix structure. That's not important for you to know. The main 5 types of collagen are what they do. Here they are right now. Type 1 is the main. It makes up 90% of your body's collagen. Densely packed and used to provide structure, skin, bone, tendons, and ligaments. It's connective tissues. Very, very dense and heavy. Then you get to type 2 is elastic, real rubbery cartilage and joint supports and so forth. It's an elastic cartilage. It's not something real hard and it's not something real soft. Number 3 is another big one. It's found in muscles, arteries, and organs. And this is the literal fabric. It's a fabric. And it's a connective tissue fabric. Type 4 is found in the layers of your skin, which is membranes again. And type 5 is found in the cornea and the skin, the hair, and the tissue of the placenta. Okay, I think I'll just leave it at this. I see no reason why there shouldn't be a viral study database on what is normal in the human body in different locations, fecal, lung, lymph nodes, where it is 100% in the body, where these things are. And then we build a database by all of those different characteristics. And then diseases, what's affiliated to these diseases. Sooner or later, they'll fall into categories. And these are central roles of enzymes as biological catalysts. Enzymes are only there if you have bacteria. Bacteria is only there if you've never destroyed it or damaged it with antibiotics, and all the things that we have in our modern lifestyle is injurious to a lot of the bacteria that is beneficial to you. So we need to start looking at this very carefully, and I don't see any reason we can't have a database like this. Something very inexpensive, not intrusive. You don't cut anybody open to get any of this stuff. We might be able to get stuff from autopsies, like lymph nodes and things like that, because those are really important. Lymph nodes are just saturated with it. And those are the dumps of all of the toxins in your body. I believe those are the source of a lot of cancers. Alright, next video I am going to concentrate on the lymph nodes and the lymphatic system and the network in your body, and how many lymph nodes there are and where they're concentrated, why they're concentrated where they are. And what about the fetal circulation and a child's bacteria? They don't have any bacteria yet, so they're on the edge. When you get older, your bacteria gets bad, you're on the edge. If you've been taking antibiotics or whatever, killing your bacteria, you're on the edge. We need to be able to figure out where is the edge. You got to stay in the middle. You can't be on the edge one side or the other. If you are, you're open for invasion. And we need to be able to see with a very simple analysis of what's in your body, not intrusive. It should be able to tell you, you're missing this, this, and this, or you're way out of the boundaries on this or that. I don't see any reason that anybody should want to go against that kind of information, collecting that kind of data. Again, I don't try to tell anybody do this or do that. I'm a researcher. And I think this is the road that this research should take right now, is to examine what is actually the human body's bacterial content. You have to know not only what the bacteria are that exist in here and there, but you have to know concentrations. Is there a lot of them there? Should there be a lot of them there? So there's a little work to do here, but I see it as a path forward.